All right. All right. Afternoon, everybody, and welcome to Open Source Antibiotics meeting on the afternoon of Friday, the 19th of February. So we have a bunch of things to talk about today, including some recent potency data and also tox data. Is there an obvious way around we should do this? Maybe tox first. Does that make sense to everybody? And then maybe Alex can present data if that's all right. You probably need privileges to do that. In which case, let me make you a co-host so that you can do that. Oops.
I just tried to share. I think it works.
Oh yeah, does it? Okay.
Can you see my screen?
No. Yes.
Yeah.
Yes.
Yeah. Okay. So
that's great.
Yep.
So if you're happy to take it away, let us know.
Yep. Okay, just start. F5. Yeah. Hello, everyone. Nice to see some faces I haven't seen before. Thanks for asking me to show you the data you've been waiting for so long. So first off, I just wanted to talk a little bit about those response curves because I'm going to be showing lots of them, and I just want to make sure that everyone is on the same page. So when we do toxicity screens, we generate this type of dose response curves where we look at the kill or viability, whichever way around you want to look at it, as a function of concentration. And typically these curves take this sigmoidal shape. That's ideally what we want to see to be able to fit the data nicely. And the way we fit the data is by using a mathematical model described by a 4-parameter logistic curve. And that is described by these parameters here. So you have the minimum effect, namely the concentration at which you see the least cell kill, the maximum effect, the maximum potency. Then we have this slope, so we can fit an asymptote to the sigmoidal curve, and we can get this slope, which is called the Hill slope, and that's important when comparing compounds, especially thinking about mechanism. And then we have something which we call the EC50, and that is the 50% of the measured effect. That is because, so ideally this curve would go from 0 to 100%. So maximum and minimum would be 0%. We have a concentration at which we have 100% kill, and we have a concentration at which we don't see any impaired growth anymore. But that's not always the case. So if for whatever reason the concentrations that we're screening are not potent enough, and this slope, this curve starts higher than 0%, then we have EC50, and that describes again 50% of the observed effect that of the observed effect. Now the CC50, which is more commonly known as the IC50, is something that the COADD protocol has looked at, and that's why I'm including it here as well, because I guess that's the figure you guys are looking for, and that is the concentration at which 50% of viability is reduced. So typically IC50 is higher than EC50, but it doesn't really make sense to talk about IC50 unless you observe 50% reduction in viability. So the way we conducted the experiment was following the protocol supplied by Dana, and I guess the way people do it for the COADD, that was in line with the compound solubility, as Dana said. So we started at 32 microgram per milliliter, we diluted 2 times, and we observed the response for 10 concentration points down from there. So this is the curve for tamoxifen, which is the control they use in the protocol. So this was my first go-to, just to make sure that what we are doing is in line with what was done before. So otherwise, nothing would be wouldn't be building on each other's work. So you can see EC50 is 11.12 microgram per milliliter. CC50 is also similar, and that's, as I said before, typically IC50 is higher than EC50, but this is within the standard deviation, so that's fine. The value quoted by the COADD protocol is 9 plus minus 2 microgram per milliliter, so I think this is good enough. Obviously, it's not exactly the same figure, but I will also talk about the fact that we used a different assay as opposed to the COADD protocol to measure the response. Now, 821, quite potent. We don't see 100% kill, but nearly there. So CC50 is 1.65 microgram per milliliter. I'm not sure what else I can comment other than giving you the values. I'm summarizing at the end, so maybe that makes more sense. So 822, again, 2.86. Again, we don't see 100% kill, so this is not a full response, but the data seems pretty consistent. 861 is one that I couldn't fit the data because yeah, it doesn't line up with the dose response behavior. So I can't calculate an IC50 because we don't observe 50% kill. So 862, same story. We don't observe 50% kill, so I guess this would be one that you're not also not interested in. 865, again, quite potent, similarly potent to 822, I guess. 2.07 microgram per milliliter. 869 is similar to tamoxifen in terms of potency, so 13.74 microgram per milliliter. We do observe 50% kill, so that's good. 870, again, quite potent, less potent than 822 and 865, but still potent. We observe 50% kill. And 871, slightly less potent than 870, but still potent enough, I guess. So this is, I guess, the summary. In the protocol, they classified the compounds as whether they have CC50 lower than 32 microgram per milliliter, which is the case for most of them, apart from 861 and 862. And then we also observe a maximum kill higher than 50% for most of them, apart from 861 and 862. So that's in line with the EC50s, the calculated EC50s and IC50s. So just briefly to comment on the assay that we use, because like I said, as I mentioned at the beginning, we used essentially a different way to detect the same thing. So the way we measure cell viability is by looking at mitochondrial activity and metabolic activity of the cell, essentially. And the way we did it is via the MTT assay, which is it gives you a colorful compound which you can measure the absorbance of, and you can associate that with viability. The reagent used by the COADD protocol is resazurin, which in turn is a fluorescent reagent. So there are some parallels to be drawn there in that the mechanism of action is similar, but obviously fluorescence is more sensitive, but on the other hand requires more specialized equipment, black plates. So overall, it would be a more expensive thing to do. Also, MTT, in terms of just classifying if IC50 is more or less than 32 microgram per mil, I can just look at this plate and tell you that my IC50 is going to be between these two concentrations. I don't even have to read it. So that's just very high throughput way to screen things if you should be so inclined. Again, as with everything, if you have it wasn't the case here, but because all compounds were colorless, so their spectra didn't interfere at any of these wavelengths that we are reading at, either for absorbance or fluorescence, but that is something to consider. So for res azurin, because it's a fluorescence-based technique, we would have to run the assay in a phenol-free medium, just to make sure that that fluorescence doesn't add any confounding. And then the reason we did MTT is because we're very familiar with it, and usually it works first off, as I promised actually. But it wasn't the case for, there were quite a lot of things to optimize with this particular cell line because it isn't adherent. So from that point of view, probably res azurin is a more practical way to do it, and the standard deviation and generally the variability of the data would be less, because you also have less washing and removing of reagents and adding things, which is fine for shorter period assays like this, but it's probably not suitable for longer term if that was something you were interested in. So just to show that I also did the res azurin for myself, just to check that we, again, the cells are similar and everything is consistent. So here I'm showing the curve. This time, this readout is the fluorescence readout. So the CC50 that I calculate is 19.74 microgram per milliliter, while again, the quoted value for in the COADD protocol is 9 plus minus 2. So again, this is within, well, the next order or the next concentration point, or within the same, but still within the same order of magnitude. So I think this is appropriate. So finally, things that I would like to do with this data is just to look a little bit more at the variability and understand where it is coming from. So do some more comprehensive statistical analysis, and also group by operator, because a lot of this data that I showed you is an aggregative, both mine and Reno's experiments. So I think discerning by that confounding variable might show different patterns. And then also transfer this analysis to R so that it's reproducible, at least computationally. And finally, I just want to thank Reno, because I'm showing you the data, but it was very much a team effort. And thank you for listening.
Okay.
And sorry for going 5 minutes over.
No, don't worry. That's fantastic. Thank you. Any immediate questions while the slides are up?
It looks like you have different hill slopes for different compounds.
Yep.
Is that an artifact, or is that real, do you think?
I don't think I can comment on that without knowing any more about the compounds. I think the hill slope is good to look at if you have similar IC50s or EC50s, but then obviously also because this screen is on the same kind of concentration pattern, in that we did 2 times dilution. Obviously, if you have compounds that are more soluble and more potent, then you can start at a much higher concentration and then do different types of dilution. So then that slope will be different. So I think the hill slope is probably something to look at when you know something about the mechanism and you have similar IC50s, or when you want to understand if assays done maybe slightly differently with different concentration reagents, you can again build on that and compare, as far as I know.
Okay.
Does that answer your question?
To some extent, yes.
So I mean, well, obviously we're seeing some fairly high toxicity with some of these compounds, and what we want to know is how that tracks with potency, that is against MRSA. I guess the 2 compounds that look like they have lower levels of toxicity were 861 and 862. Does anybody, so those would be the slides.
Yeah.
6 and 7. They appear to be lower than
So
the
can
others.
I ask, are you in? I thought you were interested in things that are toxic. So
No.
I got the way.
It's the opposite.
Yeah.
So
Okay.
these compounds are intended for treatment of bacteria.
Okay.
This obviously doesn't change your numbers.
Yeah.
No, so we're interested in compounds that are effective against MRSA and not toxic here.
Okay.
So 861 and 862 are I guess the least toxic looking of the.
Also not active, I think.
Are they are not active? Okay, so I wasn't. That's the thing we've got to do is correspond those with potency against. Does anybody happen to have that to hand, or?
Yeah, it's the 3-pyridyl and the 2 and the 4-pyridyl that we're replacing the 2 on the, I guess, southwest, we're calling it. So you know what I'm talking about, the green.
Yeah.
It's on GitHub issue number 3 as the structures of all of them. So those,
Okay.
yeah, those are not active.
Could I just wrest back control from you, Alex, of the screen share?
Stop.
Thank you. So the so that's issue number. Do you want to share, Dana, if you got it right there? Right. Okay. So yeah, so X61, X62 are those 2 crucial compounds that were synthesized to try and interrogate whether 821 is effective because of its 2-pyridyl. And it certainly looks like that's the case, right? So the 821 is definitely toxic in a way that 861 and 862 are not.
Right. I guess 869 is interesting. If I remembering that correctly, that compound is not active against MRSA. But I think, Alex, it probably was showing some toxicity, so that is interesting, and it might suggest that this mechanism we were worried with the metal chelation is not the mechanism of toxicity, right?
It's lower, 869, right? It was like 13 or something.
Yeah. So 821 is the most toxic, and then 869 is the least toxic.
Aside from 861 and 862, right? Yeah.
Exactly.
Okay.
Can I ask the differences between the compounds, Alex? Are we talking orders of magnitude, or what are we talking?
No, I mean, so it's 1, 2, 3, 5, 13, and then 2 that I couldn't calculate.
I mean, by the standards of normal cytotoxic compounds, that's not a huge difference.
Yeah, exactly.
And I'm wondering how much one should read into these, whether these distinctions are fine enough for you to come to conclusions about mechanism and so on. So and Alex, I mean, if you were going to do a statistical comparison of what is significant and not so significant, I think it may shed more light on how different they actually are, And then can I ask, what sort of toxicity do you expect?
I think there's no expectation. It's more that we hope for low.
So can I ask, why did they use tamoxifen then as a control?
Oh, this is that they use that for coad, is that right? Is that?
Yes, yeah.
Yeah, I don't know why they use that. That's just, I mean, they're a high throughput, screening facility versus the escape pathogens. I don't know why that would be there. I figured
it's important for mechanism.
So mechanistically, the compounds, where do you expect them to act?
We're trying to figure that out.
Okay.
So we've had some experiments done at UNC Chapel Hill to try and elucidate that.
They came from kinase inhibitors, so that's
originally
potentially a mechanism.
Just asking because of these assays were doing relatively short-term toxicity, or I mean, Alex will correct me if I'm wrong, but one of the concerns you might have, whether you pick up all the toxicity, in the sense that if you have compounds which would interfere with DNA replication, things like that are linked to the cell cycle, would all of those cells have had a chance to cycle and therefore notice that they've been poisoned or not? So you might get I mean, it won't get less toxic, I suppose, is the bad news. You might find more subtle differences coming out, and I suppose, as Alex alluded to, this essay is in a way non-ideal just from a number of mechanistic elements of just how it's done, and which was one of the reasons why all of this took a bit longer. And what one could consider just doing an MTT in a conventional way and see what difference comes up there, and whether those are, I mean, I guess we have some indication of where IC50s or EC50s would sit, and we could probably look to optimize the range that we cover to fall within that IC50 range, and we can see whether we can in that way get a tighter difference between the compounds. But having said that, I mean, if it's, I'm wondering how much differential we can see here and how much we can read into that is what I'm wondering about.
No, I know what you mean. I guess all along we've been concerned that the toxicity is associated with this 2-pyridine motif. So the things we were just looking at up here were the compounds that we sent to you, and it's always been this motif that's been the issue, or the worry, I suppose. Mm-hmm. Because potency falls off against bacteria if you remove that 2-pyridine motif having the nitrogen here. So these compounds are not active against MRSA. Mm-hmm. And so we've always thought that maybe this is it, and that maybe this is going to be something more generically toxic, which is the whole reason for the measurements. So it's more about whether we're seeing a correspondence between potency against MRSA and toxicity. And in these results, we kind of are, in the sense that these are definitely inactive compounds against bacteria, and they are the least toxic that we're seeing here. So it's a sort of broad class of toxicity that, rather than a neat trend. There are some other compounds here, though, that came from Laurie, who's on the call. And again, I don't know if you can see, hopefully you can see these, they're big enough. But so in these cases, we've again got, it's a slightly different measurement, and Laurie will be able to tell us, what the difference is between these and the kind of measurements we were just seeing. But again, many of these compounds have the 2-pyridine motif. And the one that doesn't, 855, is the one with the least toxicity. But we were a little bit heartened by some compounds that had the motif. So 856 here has a 2-pyridine motif, didn't look particularly toxic, but was inactive. And so I guess we were interested previously in this compound 983, which is this guy up here, which has the motif that we're worried about, but has this kind of tertiary nitrogen in this position with an alkyl group on it here. 983 wasn't particularly toxic, so we were kind of hoping that this would have activity. This may be the segue into the next bit, right? Because we now have potency data back on these. Have I got a cross wire, or is that right?
That's correct.
No, that's right.
All right. So 983 is the crucial one. So should we just quickly deviate over to those new results as well, so that we can see if this holds, because 983 is the crucial one. Whether we have an example of a compound that is not particularly toxic according to that measure. And okay. So these are they, right? You just put these up. So what was I saying? 853. What did I say? Or what was the number I just said? Sorry.
It's 983, and it's lowered down.
No, but these are the wrong compounds, right?
Yeah, that's for the efflux.
Got it. Sorry, wrong. Yeah, here you go.
Yeah, that's it. 983.
Which is potent. As potent as we've had. That guy. So these are the new results just posted from Paul on Laurie's compounds. all, you know, showing some activity, which is great.
Which is actually really nice because we can just tweak that till infinity, right? You know.
I think you want to put a few more of these through the toxicity assay to show that you have got a robust distinction.
I mean, 983 being key, I reckon.
Yeah.
983 and 979.
Yeah.
Because that also has VRE activity as well.
Uh-huh.
979, the cyclopropyl.
There were, incidentally, while we're looking at this, there were the MSSA results, which is, as I understand it, the same but lacking efflux pumps, which Paul also looked at, and the potency tracks pretty much exactly. So the conclusion from those data would be that efflux is not our problem. In other words, it's not the reason for the limitation on potency.
Which would also confirm the fact that entryway always says they pass, regardless of potency.
Right. Sorry, entryway said they all passed.
No, a pass and entryway is meeting all three criteria. These all meet 2 out of 3. They never have the primary amine. That's the third. So I think that actually that's not a pass.
But experimentally, this means they are passed, right? Because they're not being pumped out.
It means they're not substrates for that pump.
Yeah, I was wanting to ask, those pumps, how do they, in their substrate specificity, relate to mammalian cell pumps?
Sorry, how do you mean? So the entryway are referring to the efflux pumping gram-negative bacteria, right? We were wondering if the efflux pump and MRSA are pumping them out. Is that, or were you asking about something else?
Well, I'm asking whether pumping out in bacteria and pumping out in cells is in any way related in terms of substrates, or whether differential pump activity can bring differential. You know, if something can be pumped up by mammalian cells but not by bacterial cells, whether that is a route to some specificity.
I don't know the answer to that question, I'm afraid. I mean, so just looking at these latest data, I guess, in terms of, as you were just mentioning, Alex, future work, then given the broad classification we just seen in the results you have of, you know, low cytotoxicity corresponding with low activity against MRSA, these new compounds up the top here would be, if there was any more work that could be done, and that depends on bandwidth and resources and everything, then checking out the toxicity of these compounds, which we have in the lab, would be of a high priority to see if we can find the therapeutic window there, a selectivity index window.
I mean, it's possible to could we just run 979 and 983, because those were the stars of the show. Just run those two.
Yeah, and also, I mean, 2 or 3, I would guess, just if we can have something which is inactive and something which is active. They're all pretty active, right? But yeah, we try and cover the spectrum, I suppose. Just going back. Yeah.
So do you have any golden compounds or like something? So tamoxifen, because I thought tamoxifen is important for mechanism, so that's why I wanted to make sure that that's all in line. But do you have anything that is commercially available or something that we can benchmark? I think that would be useful.
Yeah, unfortunately, we don't know the mechanism of the toxicity. Otherwise, you know, we could suggest a positive control.
I mean, yeah, but I mean, I think that the whole point about the using the same compound, though, right, was that we would get the similar. We would have confidence that even if there were assay differences, that that we.
Yeah. No, that. But I mean, in terms of, I guess if you wanted to be able to discern better.
We don't have any information on the MOA or MOT for that matter.
I mean, the two compounds we looked at are quite different now, top and bottom, isn't it, structurally?
Well,
they have the motifs that you're
Yeah.
interested in, but otherwise they're really quite different, isn't it?
well, they have this link here, which we didn't initially have
yeah.
with this kind of compound. This is our standard 821. So yeah, they've got this little bent core here, but the rest of it is broadly similar. You're worried that we may be of mechanism hopped.
Yeah, so yeah, essentially, but I mean, don't know. It's not my area. So.
Yeah. But does it matter, Matt, if we've got a
No.
new mechanism, as long as they're antibiotics?
Yeah, phenotypic drug discovery forever.
Yeah.
Yeah, I mean, I guess what I wasn't sure about is in terms of trying to discern toxicity of particular moieties compared to others. It's quite a big jump, isn't it, from bottom to top?
I mean, could we not try making 893 but lop off the bottom two pyridyl?
Yeah, I mean, I would want to just double check these first. So 980 is fairly toxic according to this assay, which is remarkably similar to the one we're looking at with different alkyl chain on here. I mean, I definitely want to get these checked out again.
I think that one, so 980 actually has a methoxy on the imidazopyridine core.
There.
Upper position.
There.
Yeah.
Oh,
I
yeah.
think the structure's wrong. Dana was just going to double
That is
check.
Sorry,
correct.
that's
it
my
has
fault.
a methoxy, yeah.
Yeah.
Sorry, guys. Sorry.
No, that's all right. There it is. Yeah, right.
Yeah.
Okay.
And so we have seen differential toxicity. I think that's the position that's normally that one is better than the substitution patent on 855, but I would need to go back. Like, there's a lot changed between those two. I'd need to go
Well,
back.
the good news is it doesn't seem like we need a substituted core.
No, you don't. Well, it doesn't look like that from this data.
Okay.
Sorry. Yeah.
No, so then it's 981, 982, 983 because that's the jump tenfold.
Yeah, and so we have PMM data on 983 as well, on all of these. And there's also a big jump in terms of PMM data on 983. It's 23 micromolar versus single-digit nanomolar single-digit micromolar for the rest.
Okay, I mean, it's really interesting that you get these differences with these tiny.
Yeah, well, I
alkyl.
find the difference between 983 and 979 quite striking.
Well, I mean, the one where you just showed a tenfold jump. If you can talk me through that one again, yeah.
So yeah, you're going here from like
983 or 981, 982, 983.
yeah, so you're going from 5 to 50.
So that and structurally,
I don't have an explanation for you in terms of that one. I mean, we also see it like that's what I'm saying. We also see it in a PMM cell line, and I can't explain it.
So Andreas, yeah, structurally, it's just a modification of this alkyl group.
So, and which one was the more toxic one?
These are the more.
Yeah.
This
And
is the last.
solubility is not an issue there.
It's not amazing. Matt, would you mind if I share my screen for a second?
Yeah, go for it.
So I added the host cell line, sorry, not host cell line, the toxicity cell lines that we have, as well as ADME data for these compounds. And 983 is definitely the pick of the bunch, in my personal opinion, when you consider host cell toxicity. But no, I mean, it's really—
These are calculated log, these, yeah?
This is measured at pH 7.4.
Okay.
And the solubility is measured?
Yes, it's thermodynamic solubility.
Huh. I mean, it's not the worst. 983 is not the worst, but it's not great, but it's not the worst.
It's not great. Yeah. So we have other analogs at the moment. The chemist here, Quillen, is actually making some other derivatives where we've kind of got primary alcohol on sort of the alkyl chain. So they could actually be quite interesting. We're hoping that will help in terms of solubility. They'll probably give us
Another thing I was thinking of was amide or amide functionality. We start with a acyl chloride and then attach it to it. Sort of like chlorobutanoic acid or something, and then attach the chains. I'll draw up a couple ideas I've had, if that makes sense.
I mean, if the implication is from these combined that we should be pursuing this core more because there has a better chance of selectivity, but we want to get them checked.
Yeah.
I agree totally. I think the 5+6 core is probably going to be easier on all of us and probably easier to make too, but we do need to verify before we hop.
Because this is interesting because of course it comes back to what we were talking about last time with Lee and the mechanism of action studies, that if we had things that were generically toxic, then it's less interesting for him to do the experiment on because he's using MRSA, lysate, and you know, maybe we're using compounds that are just generically toxic, and so well, I guess it doesn't make that much difference. So, but if we had a compound here that we were more happy about the selectivity index for, then that might give us more meaningful data for the mechanism of action stuff.
I think that's absolutely correct.
So if 983 holds up, then that will be a better compound to use for the repeat of those experiments. Does that make sense?
And as we're making more of these derivatives, we can certainly include a like set aside a sample to ship to you as well if you're interested. Cool.
I mean, yeah, we don't want to, you know, interfere with whatever you're doing, but if there's common chemical space that we can work on.
Yeah, it is. Yeah, this is kind of the area that we're pursuing at the moment, and then we're also substituting off the pyridol as well.
Very interesting. Well, that's very useful. I mean, yeah, it is important that we try and prove that. So we have sample left, right, from you, Laurie, in our lab. Dana, we've still got sample from Laurie left, right? We didn't consume all those donated samples for the.
Well, I did dissolve it all for Paul, but I don't think he uses all of it in his assay. So I can check with him and to see if he has some stock solution left.
Was that DMSO?
Yeah.
Okay. Alex, how do you do the assay? Do you take DMSO samples?
So I dissolved everything in DMSO at 3.2 milligram per mil and diluted 100 times from that, and that's the starting point of the curve.
Okay.
Twofold. Yeah.
Okay. I mean, so you're busy with everything else, but can we put our request in your bandwidth queue to see if we can have one or two compounds extra measured?
Yeah, I think we're now better what we're doing, so it shouldn't be that big of a
I'm wondering, do you know how tedious is the assay compared with the working with adherent cell lines and because I mean at the moment it seems the only reason for this assay in this form is that it compares to something someone else has done elsewhere. Mm-hmm. There's no mechanistic underlying reason that says this is showing either in the cell line or how you do the assay. And I mean, if you wanted to compare things directly, you could choose any sort of toxicity assay you would like to use, isn't it?
Yeah, I think that's right. I think all you need to do is, from our point of view, is to take a handful of these and do a sort of bridging one where you do them both in both assays and show that you get parallel results. And
yeah, I mean, just from my very limited experience with trying to develop antibacterial agents, and often a sort of one point where we've seen them acting on is sort of interfering or binding DNA and not necessarily super specific. And so I'm wondering whether just sort of doing a bridging with some of the compounds where we looked at, where we just now looked at these pyridine segments and then your others and putting that into a MTT that runs over the 7 days, Alex, it may show more differential between the compounds, and if that's the case, then maybe it gives you better guidance of where to go.
And which cell line?
Well, I mean, you could be any, I suppose. Really, it doesn't matter too much. I mean, adherent, obviously, I would go for.
Yeah, I mean, I just mean it should be human, right? Because otherwise.
Yeah.
Yep.
Yeah.
Well, probably doesn't make much difference with mammalian, but yeah.
Okay.
You have more justification to do not to use mammalian, so yes.
What's your preferred assay that would be operationally simpler?
Well, I mean, it comes obviously. Typically, we look at cytotoxicity, trying to find it, then when we have it, then you would maybe look at compare different cell lines which have a different genetic makeup, and then that gives you a hint about where you see more toxicity, so we don't have a particular preference. Anything that's easy to work with for initial screen is fine, I would say.
So if I'm not mistaken, you're looking for cancer drugs then? Typically,
yes. And I mean, I don't know whether we've got the fibroblasts kicking around, WI38. We used to have those.
Yeah, we do have them.
Fibroblasts would be good because most MRSA infections start through the skin.
Yeah.
It's not something I've worked with a lot, but yeah.
They should be very well behaved, actually. Okay. So shouldn't make much difference. Or, I mean, if it's quick, we could use anyone. I mean, if you know, there's direct comparison between compounds that I don't know, if you have a short list of 6 or something, and you want to see how they all compare in the same assay, then that's easy. If continuity is more important, then I think working with what about changing to resourcing? Still a pain, isn't it? The assay.
You mean Resazurin?
Yeah.
Yeah, I mean, no, we can do it. It's again, we can do it, but as I outlined, there are parallels to be.
Well, I mean, you spend a lot of time actually on it, isn't it? Comparatively to MTT.
I mean, I don't know if we want to do the week-long protocol where we try to discern like some more subtle differences. I don't know how, because obviously fluorescence is different to read out, so then we have to again optimize cell number possibly.
Yes, I mean, so the question starts in a way with a question of how discerning is this assay, and is it discerning enough to want to draw conclusions about what structures might carry toxicity. And if that's for those compounds that we've looked at, something you still want to pursue, then it might make sense to do another assay, slightly different form with those compounds. If, on the other hand, your thinking is now that, well, maybe this other structure with the fluor and the different length alkyne chain is to be explored, well, it's a slightly different scenario, I think, and it comes a bit down to your priorities in this.
I think the first priority is to identify whether we have a selectivity index. So we have compounds that are potent against MRSA and not cytotoxic. If that's the case, if that's not the case, then we have to abandon ship, publish, and get out. If it is the case, and we can explore a compound that gives us that index, then yeah, the next thing is to say, well, it would be great to know what the mechanism of action is. Intellectually, you want to know what it is. for sure, but just being a little bit pragmatic, you don't want to pursue a series which has no future, if you can avoid it. But if it is the case that we have a selectivity index, then we definitely want to know what the mechanism of action is. And if there are clues that can be derived from what kind of toxicity we're seeing, that would be extremely useful.
So, I mean, when I look at those numbers, my eyes glaze over very easily. There's different IC50s and EC50s, and an order of magnitude is easily lost on me. And I was wondering whether you've tried to put this graphically in a way where you sort of see a correlation between both. And I personally would find it much easier to assess, but that's just me not being great with numbers, I suppose. But
no, sure, that would be. I mean, I think that's what we're all doing in our minds, is creating that graphic on the fly.
Yeah, normally I come a cropper doing that. So.
But I mean, at the moment, we do then have data from 3 different toxicity assays, slightly different toxicity assays. So Laurie's assay is slightly different from the COADS one, and we've got another one again. Introducing a 4th one, we have to be really sure that we have those bridging compounds that have been tested in all assays to make sure that the one diagram can take all of those data points and we're seeing something that's real.
But how, I mean, those different assays, how different are the numbers that come out? I mean, as I said, I don't have a good feel for it, but you know, with cytotoxicity under cancer compounds, I mean, we normally do 1 in 10 dilutions, and if it isn't 10 times more, it's basically the same, you know. This is how discerning I think these assays are. Yeah, so that maybe if you could argue that if they are sort of in the same ballpark or similar ballpark, you could still at least to get a feel for things, because I as I said, I find it difficult to get a feel for things by just looking at the numbers. Put it all together and see what you get. I mean, if on the other hand the differences are big, well, I guess it's good in the sense that it's discerning, but I just don't have a feel. I don't, you know, I can't pick that up that quickly.
I think, I mean, again, the number that people want to see in this area would be that selectivity index. So the toxicity difference between MRSA and mammalian. And you'd expect to want to see about a 50-fold difference if you're going to have something of value. So, you know, 1 microgram per mil toxicity versus a 50 1 microgram per mil potency versus for sort of 50 something for mammalian. You know, you want those kinds of numbers, I think. And if they get much closer than that, you get a bit nervous.
I think at the moment, though, you just want some evidence that the 2 things are divergent.
Yeah, that's right. That's right. Yes.
And do you, off the top of your head, know which compounds are most divergent? I mean, because there's absolute activity and then there's divergence. And I mean, I just by looking at these numbers, I cannot get that so quickly in my head what's what, you know. And if you're looking for structural clues, you might you know, try and create a sort of ad hoc index like that, and maybe see whether that structurally guides you in a different direction from just looking at it as you have.
Well, the best were the compounds we were just looking at. I think those had the best, the biggest difference between the 2 potencies.
Okay.
Yeah.
Yeah, I think we need to test 1 or 2 more of those and just see whether
Also,
finding.
your benzimidazole structure hop is in the pipeline, right?
Yep. Sure. Yeah. So there's some. We don't have time to talk about it now, I think, because we're coming up on the hour. But there is chemistry in the pipeline that also diverges a little bit more too. Yeah. Okay. Well, it's been very useful. So I guess
been really good.
Sorry, say again.
I'd say it's been really good.
Yeah. So, I mean, first of all, thank you so much for getting the data. That's absolutely crucial for us. So that's really been very helpful. And thanks also in his absence to Paul for getting the data back on the compounds. So we only gave him like 3 days ago, right? 2 days ago. So yeah, his assay's fast to get the potency on against the bugs. And I guess we have an action item to follow up with you guys on evaluating a few more compounds so we can be sure what we're doing. And those are going to be pretty key experiments. We haven't had time to talk about it today, but yeah, I mean, the chemistry is in the works, and Giada has been looking at mechanisms of action stuff as well. And there's a GitHub issue on that where she's been trying to find structures of PDB files, crystal structures of that list of proteins that Lee Graves gave us last time. She's been trying to find crystal structures of those, potentially with things bound, to give us a sense of whether or not things that are bound may be similar in appearance to the molecules we're looking at. You know, whether there's any clues out in the literature for ways in which our compound may be interacting with proteins. So she's beavering away on that, which is great. But we haven't got time to talk about it, I'm afraid, I don't think. So we'll have to save it till next time. Was there anything else anyone
Bit of a
wanted to know?
cliffhanger.
Yes.
Can I just ask? Can I ask a question?
Sure, yeah, please.
Yeah, sorry. Because you mentioned that you have data, well, I'm guessing similar data, toxicity data on different cell lines. Is it possible for me to see that, or is like just so to get an understanding, or like did I misunderstand that?
Toxicity data on different cell lines.
Or
Yeah, I'll send you, Alex.
okay, yeah. Cool.
Great.
Thank you.
Well, nice meeting everyone.
Yeah, thanks for coming along. You know,
All right.
always welcome.
Thanks, Alex.
Yeah,
Thanks,
great
everyone.
stuff.
Thanks,
And
Lino.
see everybody next time.
Thanks very much.
Bye-bye.
Bye bye.
Bye.
Thank you. Bye.
Bye.